Okay, we're back, and I'm about to introduce Shuguang Zhang, but let me first say this is the afternoon session of the inaugural Osmocosm conference about machine olfaction technologies for the planet. We are so thankful to the organizing team and to the panelists and to the speakers and to the audience for providing us with such great content, such great questions. It is our first time and we're learning as we're doing it, and we've learned so much. It is my distinct pleasure to, Brian, I think we're having someone leak in. Please, Dr. Shuguang Zhang will talk next. And let me first set the stage for Shuguang, because it is he is my mentor, and he's the reason I'm at MIT. He brought me to MIT in 2004 in a very crazy phone call he made to me once while I was busy typing, finishing my PhD thesis in Texas A&M University. I suddenly get this phone call. This was before smartphones were everywhere, and it says MIT on the caller ID, if you remember those LCD things. And well, I had to pick up, and then he says, la 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 la, you have to come, you have to become a postdoc with Crick, Francis Crick. And I knew, of course, of Francis Crick, but I didn't understand why that was the case. And but anyway, he explained it to me. Unfortunately, Francis Crick died. So I went to Caltech, met with his postdoc, and then I also came to visit the lab of Shuguang Zhang at MIT. And of course, I fell in love with that lab. It was back then called the Laboratory for Molecular Self-Assembly. And 17 years later, here we are building noses. And we've gone through so much together, from scuba diving for seaweed to make solar panels, to going to Japan and elsewhere in crazy places, getting into adventures. There's a video out there somewhere of Shuguang videotaping an exploding volcano and Volkswagen-sized rocks raining on him. He is sort of the Indiana Jones of the science world in many ways. He calls me fearless, but I think he's even more fearless than me. So what he has done lately is absolutely staggeringly important for the world of pharmaceuticals, for the world of membrane protein science, and for the world of nose makers. So as you remember from my lecture, perhaps, and from many others, we told you that the olfactory receptors are membrane proteins. That means they have to sit in between water, and the middle part is a lipid bilayer, a hydrophobic layer. Well, that is a problem for many technologies that aim to decipher what goes on with these receptors, because the membrane environment is kind of finicky. Once you take the protein outside the membrane environment, it's difficult to stabilize it. And Shuguang, of course, is historic in his discovery of peptides that are capable of doing this, of stabilizing, going in there to the hydrophobic pockets on the outside and sort of stuffing it in there to make sure that the guts of the protein don't spill out. But even more staggering and more important is that he has created a code called the QT code, which spells a whole new era of pharmaceutical development as well as nose making. The QT code is something which she will talk to you about, but it's just almost a magical technology. It's been confirmed by many other people that it is possible to, using this code, to exchange up to 15% of the amino acids in such a way that the previously membrane-bound proteins become soluble. This is some, it sounds like some nerdy little detail, but it's not at all nerdy little detail. It's like giving this protein the ability to be produced en masse in solution, opening the door to massive throughput assays for biopharmaceuticals. It is an incredible discovery. It deserves your attention, and it is the future. With this, I give Shuguang the floor. Please take it away. You have 15 minutes.
Okay. Thank you. Thank you, Andreas, for such an introduction. Here, Andreas and I both work in the media lab. This building was designed by I.M. Pei. Talk about innovation, this was in the 1980s. The building encoded code MIT. Can you see? M here, IT here. That's the code designed into the building. And both Andreas and I work in the same building. As Andreas explained to you, the olfactory receptor is the key for our smell, for distinguishing different odors, and for the key for the dogs to smell cancer and non-cancer. The key right here is the olfactory receptor. Andreas said the olfactory receptor embedded in the membrane system. If you take them out from the cell membrane, system is right on the nerve system, on the neuron here in your nose. And if you take them out, then become aggregated, then lose function very fast in a few minutes. But interestingly, nature made the same kind, two different kind of proteins. One is a totally water-soluble hemoglobin in our red blood cells, 30% solubility in our red blood cells for the alpha helices. The second is a GPCR, including nose, olfactory receptors. This protein is totally water insoluble. How nature makes the same kind of structure alpha helices? One is water insoluble, water soluble, the other is water insoluble. The question is, can you convert them? I busy at the time in 2011. But nature has done it, as you can see. So we should be able to do that as well. Here is a textbook, 1999, first edition published in 1992, shows in nature again, the three alpha helices. One is hydrophobic, one is half hydrophobic, third one is hydrophilic, means water soluble. Then have the same structure. That's the key, okay? And here is a structure 1.5 angstrom per amino acid race, 100 amino acids per turn, 300.6 amino acids, 360 degree, 5.4 angstrom per helical turn. They are identical, okay? That's very important to remember. Again, here is 20 amino acids in our body, in life, all life on the planet Earth. That's why coronavirus can infect the human and we die from it. Also, you can eat a piece of steak, you don't become a cow. That's for the same reason. As also bacteria can produce human protein for us, insulin included. So in this 20 amino acids, there's some very important, interesting pairs you can see. This pair here is one is valine, it's totally water insoluble amino acid. This one is a serine, it's a water soluble protein, amino acid. Here is again leucine, water insoluble. This aspartate, asparagine, glutamate, and glutamine are water soluble because the red oxygen, blue nitrogen. The idea is, can you replace them? Replace this one with valine, and also replace leucine, and finally the phenylalanine and the tyrosine. Can you replace this phenylalanine with water soluble tyrosine? That is the code. So it's more in detail how the bind to the water is showing here. God is in details. Okay, here is God in details at the molecular level. This molecule side chains can surround by four water molecule. That's why this amino acid is water soluble. This molecule, water side chains, can surround by 3 water molecules. That's why this amino acid is water soluble. That's it. Okay, if they change to green, then water insoluble. That's the reason for we can make water soluble membrane protein olfactory receptors. Leonardo da Vinci said, simplicity is ultimate sophistication. Okay, here is the QTY code here, showing there's a QTY, the QTY replaces LIVF. The L is replaced by either N or Q, we select the Q, or the I and V replaced by T because similarity of structure. That's the key, similarity of structure. Finally, phenylalanine replaced by tyrosine. Before replacement, you can see this helical well, all the green are water insoluble, showing on the surface of those protein olfactory receptor right here, called OR52D1. It's full of the hydrophobic green, yellow part is water insoluble. Now we replace them with water soluble amino acids as QTY. Now the olfactory receptor become water soluble. That is as simple as that. That's what we call the QTY code. you can see there's two amino acids, two receptors. One, the green, is a native structure. Cyan is a QTY variant analog structure, then nearly superimposable. The differences is 0.68 angstrom difference. There's another one. This green is water-insoluble native protein, and cyan is water-soluble analog. Again, the differences is 0.71 angstrom. Remarkable. This explains. Okay, so before this with a glove, it's water-insoluble version, native one, version is hydrophobic version, water-insoluble. But the function of this is hold a pen, is doctor hold a knife for doing surgery. And the people make chip makers, making Intel chip computer and the iPhone, the chips, they must wear gloves, okay, to avoid contamination. So that is called water-insoluble hydrophobic version. If it takes out, this water-soluble version will not hinder this capability. Now, in order to making something useful for the make water receptor, water-soluble olfactory receptor, you need an extremely high density on the surface, like a dog. You saw the video. The dog has how many 100 times more olfactory receptor in the dog nose than human. And that's because of the surface area. So now we take another technology combined from bacteria surface protein called acellular protein. We can now place them on a silicon wafer, you can see. And then you can use high resolution imaging. You can see like a 2-dimensional crystal, like a tile, like a kitchen tile, or like the wall tile is a tile. Each square is precisely measured. In this case, it's a 13 nanometer. Okay, it's 3,000 times thinner than a single human hair. So now the idea is takes protein out from the membrane and then make it into water-soluble version and coupled onto another protein called FC domain of antibody. Now this protein now is able to couple onto acellular protein, form a 2-dimensional array, 4 trillion molecules per square centimeter. That's how density is, okay? It's the one with the thumbnail. You can have 4 trillion molecules there. So that's why high density can do the sensing. And so this first experiment showing here, if you take a pure gold, give this signal, it will measure voltage and, and current. So this acellular alone, the red one, the green one is ligand alone, and the blue one is this one with receptor. So without ligand, it doesn't have the green. Add the ligand, you get additional signal. So this is specific interactions. That's without with different ligand that does not give this signal. So this is the first demonstration that this purified, this water-soluble receptors can be used to making a sensor for detection eventually. So there's still a lot of work to do, but we're now starting to tease out the complexity of smell without the user's brain. Use electronic interface to make a smell sensor. That's it, okay? That's the idea. We want to eventually incorporate the new device into our mobile phone, and Andreas already doing this kind of work. So we continue this research. So my mentor, as Andrea said, we come from a generation of scientific lineage. This is my mentor, Alexander Rich. He was Francis Crick, James Watson as a colleague in 1955, the taking in Francis' house, the golden helix. This is alpha helix. This alpha helix is one I'm working on now. You can have water-soluble alpha helix or water-insoluble helix. We converted water-insoluble helix into water-soluble helix. So now Andreas also comes from the genetic lineage, scientific genetic from Alexander Rich, Francis Crick. Okay, thank you very much.
Wow, thank you, Shuguang. That was a lot faster, and thank you for being on time. We will have many questions for you. Please stay for the end of the hour. Now I'm having some connectivity issues here, and if I do drop off, please, Carolina will take over. But so far, so good. We're going on to the next speaker. All the panelists will have a chance to be asked questions at the end. Please stay towards the end. So my next speaker, our next speaker is Dr. Ilya Kolb. He's a senior project manager at Aromics, and he'll tell us also about how he is expressing and stabilizing olfactory receptors en masse. Shuguang will hear it, and probably you don't know that they've done over 100 of them. With Shuguang, we did about 12 in the lab. And Aromics is a company that is very interesting. They are focused on flavor and olfaction and taste. However, I think they are slowly seeing the light that the real money and the real impact is in health. And I think that their technology is absolutely crucial. They are the highest expressing company on the planet right now. They have the most olfactory receptors. And I think their contribution will be increasing in impactfulness as we go forward. Ilya, you have 5 minutes. Take it away.
All right. Thank you very much, Andreas, for inviting me. And thank you to all the speakers. I learned a ton throughout these multiple days. So my name is Ilya. I work at Aromics. And what I'm going to talk to you about today is how we're kind of leveraging olfaction and taste, so things that we've been talking about, in order to actually help companies develop better products for their customers and give them, and how Aromics is able to give them actionable insights into their products. So in order to do that, I'm going to kind of introduce a kind of a use case or a customer case. 1 second. So in this case, we had a customer, which was a multinational beverage company, a multi-million-dollar company. And what they were trying to do is essentially release a sugar-free beverage that had the same smell and taste as the full sugar beverage. So of course, they want to make a healthier version of the product. And what they came to us with, their kind of challenge to us, was to try to measure whether the artificial sweetener can recapitulate the flavor profile of the full sugar beverage. And so one of the kind of challenges of this is that, of course, the beverage that they gave us is a very complex one. So it's not, just sugar or just 2 or 3 different chemicals. It's possibly hundreds and thousands of chemicals. And so the way that Aromics approaches this situation, as well as all of the other customer requests that we have, is it was actually brought up very powerfully by Dr. Zhang earlier, so I don't have to do too much work in terms of the science. But basically, we take the receptors that humans use to smell and taste, so the olfactory and taste receptors, and we grow them on a dish, we express them, and we can assay whatever samples our customers send us. So whether that's a perfume or a beverage, or we've also, as Andreas mentioned, we've worked a lot with some other samples in the medical space. But what we do is we flow them over our assay, and then we can see which receptors are lighting up. And so in this particular case, what we wanted to do is give this customer insights into this problem of making the sugar-free beverage. And what we're able to kind of give to them is a couple different things. So first, as I mentioned, of course, we know the receptors that are going to light up differentially in response to the sugary beverage and the sugar-free one. But also, we're able to give them the actual identities of the chemicals that might bind to those receptors. We can also, once we know the particular receptor activation pattern, we can connect that pattern to word descriptors that the company itself uses in their own marketing material. And then, of course, we can use AI and data science in order to come up with better formulations of the product. So let me jump into the data here and straight to the conclusion. So we ran these two samples through our assay, and these are kind of the results that we got. So what I'm showing here are the two samples run across 130 olfactory receptors, and these are ordered by their discriminability. So at the top are the receptors that are maximally differentiating between the sweetener product and the sugar product, and all the way at the bottom, you can see the two colors are about the same. Those are the receptors that are basically firing the same in response to those beverages. So if we kind of zoom into the first several or four of these receptors, we can see that there's at least two that are responding much higher to the sugar beverage than the sweetener one. And connecting that back to the insights that we provide to our customers, what we can say is, okay, here are the two receptors, but then also what are the target ligands that this company can use to try to get the sweetener beverage to taste and smell more like the sugary one? And also, what are the word descriptors that, for example, are encoded by this AR1, which in this case happen to be things like sweet and floral, which of course makes sense given the context of the sample. And then finally, what we can start to do, and this is still kind of a work in progress, but ultimately the customer wants to develop a healthier alternative to a sugary beverage. And so this is something where we can use our AI and data science to figure out what formulations are most likely to do that. And so just to wrap up, with our progress in 2021, so what we're kind of exploring are three different things. And the first one is actually a theme that we've heard kind of throughout this conference, which is that we know that smells and tastes in different areas of the world are perceived differently. And we think that that might have something to do with the actual genetic variation that underlies the genetic variation in the receptors. Of course, there's also other events like cultural phenomenon as well, but we think that it could go as low as the genetic variation within different populations. And so we think that that can be a guide to how we can market and formulate products for certain areas of the globe. We're also, of course, growing the database of our receptor ligand mapping. So filling out the database to have more receptors, more ligands that bind those receptors to give, again, our customers better insights. And finally, we're scaling up. We have had a lot of demand from companies to screen more and more things. And so, of course, we want to keep up with that. And as we do that, we want to make sure that we get really high quality data out. So with that, I will finish up and thank you.
That was very, very wonderful. And so many questions I have for you. We will please wait until the end. Ilya, that was fantastic and very pithy. And I'm so glad that Aromics and you are here. So for our next speaker, we have our very own, I want to say, Dr. Wen-Yee Lee, Professor Wen-Yee Lee from University of Texas at El Paso. Can you highlight yourself, Wen-Yee? There she, oh, nope. You are somewhere, I can't see you right now. Can you unmute yourself? Uh-oh. Brian, if you can see Wen-Yee, please unmute her, she's next. Oh, she says, I don't have access. Matt, could you please fix that? Wen-Yee should be promoted to panelist. She might have clicked on the wrong link.
Taking care of it.
Just texted. While she's setting up, I'll tell you that she is part of the team that is behind Sentient AI. Let me see, where is Wen-Yee? Maybe I can do it for you. And she was also part of our group that is behind the +1 paper. And I would very much like her to talk to us about how she's detecting prostate cancer using GCMS. There she is. Okay. Now, it is my distinct pleasure to bring her in. She is a co-founder of the company called Sentient, which will use GCMS to not only detect prostate cancer better than anything out there, but also do something that no other technique can do, which is bin it by a Gleason score. This talk of hers will be about perhaps something else, her work that led up to this. And I want to say one of the things that impressed me the most about Wen-Yee when I first met her through the Prostate Cancer Foundation was her poster. So in science, we tend to be very conservative. And both Shuguang and Wen-Yee are the opposite of that. They're adventurous and they're bold. And when they know that something is right, they are not shy. So her poster was so very visible because instead of having tiny little letters that you had to come this close to look at, she had written in giant letters 92%. And you could see that from a mile away. So of course, everybody made a beeline for her poster to learn what is it that 92% that she's advertising. And indeed, she had a technique for diagnosing prostate cancer that is better than anything out there. So please, Wen-Yee, take it away for 5 minutes.
Okay. Hi, everybody, and thank you for having me. So just jump to the topic. Again, this research is inspired by Doc. So I keep saying that if Doc can do it, can we do better? And I want the answer to be yes, of course, because we're human. So let's see how it goes. Okay. So I think from the panel, we already know that early detection for cancer can save lives. So I think we all agree on it. And listen to Adam earlier, we also confirmed this idea. However, if the detection is too expensive, or if the detection is inaccurate, it can lead to problems, health disparity, for instance, or is a waste of resources. And so using prostate cancer as an example, okay, I don't want to reiterate what Adam just saying, but very briefly, we know prostate cancer, using that as example, we have early detection using PSA. If PSA show up some like problem, then it go to a further diagnosis, and which is the imaging guided biopsy. The problem is that this process from early screening to further screening is wrong 60% or even higher at the time. Okay, so I'm using some number to show you the seriousness of the problem. So I give you the diagram that put out by the Preventative Service Task Force. And so capitalize this number. I'm not talking about the recommendation, but I'm using the number that based on what the data is showing, the PSA, the current gold standard, PSA only have 42% of accuracy. And therefore, it results in a 58% of unnecessary biopsy. So what does that mean? So relatively speaking, right now, say we have 20 million PSA done annually in United States only. That will result in 2.8 million unnecessary biopsies. So what does that mean? That means it's a $7 billion wasted right there. And therefore, using this idea, we know that we need a better alternative for cancer diagnosis. And from previous talks, we know dogs are amazing, right? So I show you some of the references that dog can smell cancer, prostate cancer, bladder cancer, colon cancer, and so on. Using prostate cancer as an example, the sensitivity and specificity can go from 91% or as high as almost 100%. So now what we're thinking is that what dog is sensing, the smell, is triggered by some special compound. And since it's in the air, okay, so we were going to call it volatile organic compound, VOC. And we have used this term throughout this conference a lot. And maybe those VOCs can be biomarkers. So dogs use their smell, and we chemists, we are going to use the instruments. And this instruments is what we've been talking about, gas chromatography and mass spectroscopy. We call it GCMS. So how do we do that? Okay, so I'm gonna give you a very quick run about how we do the analysis. So we go in 3 steps, simple step. One is we take the urine sample and we use extraction methods that I'm gonna go in a little bit later, called stir-bar sorted extraction. And then from there, we are able to detect what compound is in the urine, and then we're gonna do data mining and then screen, okay? So allow me to say that, therefore, our goal is that if you can pee in the cup, then we hope that we can develop a non-invasive, fast, and accurate diagnostic tool. So why can we know? Urine is non-invasive, everybody agree. And why do we say it's fast? So let me spend a little time about our analytical process. So we use a stir-bar sorted extraction. So now if you have a cup of urine and we take some 1 milliliter out, put it in a vial, and so we just let it stir. And so this stir bar, special stir bar, can pick up the metabolites, and then after a certain time, we can do the extraction, concentration, and then clean up at the same time. What do I mean clean up? There are some samples that say it's still water soluble, and this could be protein or it could be some amino acid. And these are the compounds that we decide not to look into just yet, okay? So then after a certain time, we take it out, we put it into a thermal desorption unit, and then we desorb it, and then all the VOC can get dissolved, and then we will be able to analyze on the GCMS. Okay, so this is our setup right here. And so with that being said, then we will be able to get the raw data, which is a chromatogram, and then through the NIST library, we can identify what they are. And so right now we're using very traditional searching Wilcoxon-Ransom test to fish out the significant VOC, and then we use logistic regression model to develop this model. So very briefly, that is what we have right now. We use this approach to develop a prostate cancer diagnostic model, and this is the outcome here. Right now, we are able to accomplish AUC value of 0.92, and when we do it on the testing group, is 0.86. We can also do the risk assessment. Risk, right now, we put it in a very vague group. One is Gleason score 6, and then another group is 7 and above. In other words, one is clinically insignificant per se. Another group is more as clinically significant. And because we don't have enough sample just yet, so at this point, we're getting more now, but the data is not out yet. So at this point, our training group is giving us 0.86 AUC value. And we can do the same thing for other cancer. Kidney cancer, for instance. So we have tried it on the kidney cancer. There are so many different types of kidney cancer. One type is called clear cell. And so right now we are able to show a 0.87 AUC value in the training, and then 0.81 in the testing. Okay, so, and this is a recent one. Andreas, very convenient with, very familiar with this graph. Recent collaboration with Steve using AI, we're able to use the binning concept that this approach, just by looking at the chromatogram, may allow us to really see different Gleason score, 6, 7, 8, or 9. And so this is just a graph that I want to show you that using urinary metabolites really do have the future to do a liquid biopsy. Of course, we know that the journey goes on. Yeah, we are not done yet. And so with that being said, really inspired by this conference, we know in the future, you've got to be a collaboration between among researchers, educators, clinicians, and now I know artists, for instance, and then also entrepreneurs. And I think that together, I really like PCF's slogan. Together, I think we really have a very good shot at overcoming cancer. And that's it.
Thank you, Professor Lee. What a great presentation. I have so many questions, and they're pouring in online also. Please stay on until the end so that we're going to do an extended Q&A. We're blessed with a little bit extra time today. Next up is Dr. Joseph Azzarelli. He is from MIT originally, and he went through Tim Swager's group here, and he's going to talk to us about distributed olfaction for augmented awareness for 5 minutes, and then we're going to bring him back for the Q&A. Take it away, Dr. Azzarelli.
Thank you. And can you hear me okay?
We can hear you fine, and we can see your screen. Yeah, very good.
Okay. Okay, so as Andreas mentioned, I'll be discussing distributed olfaction for augmented awareness. Before I get started, I want to echo what Aaron, I believe at OVP said yesterday, which is this is just the conference I've been waiting for. It's really been phenomenal. So I want to kind of start to ground the audience with, what do we mean by augmented awareness? So let's sort of start with this scenario, this hypothetical scenario where you have an individual, or for that matter, another entity. It could be an AI, it could be a business, and they want to interpret something about reality. And there's this information, as we've learned through this conference, in those VOC profiles, there's this chemical information that is contained in some sort of sample. But you have this scenario where the interpreter does not have access to that sample, some sort of impermeable barrier, however you want to interpret that, but it's not accessible. And so the interpreter has a multitude of interpretations of their reality or their scenario that they can come up with. And they have a certain amount of confidence about which one is the correct one, or how it's going to be perceived. Now, we contrast this with this alternative option where somehow that olfactory information is accessible with a device. And that information can then be contextualized. That's an extremely important point. Contextualized olfactory information delivered to the interpreter. And the interpreter now, with this new contextualized information, olfactory information, is able to reassign their confidence in their interpretation of their scenario or their reality. And that, of course, can then substantially influence their decision-making. And so I like to think about augmented awareness as the ability to improve interpretation confidence with contextualized information. And then importantly, that's not accessible without a device. That's the augmented part, because if we could just automatically have that natively, so to speak, it wouldn't be augmented, but it's this additional information we can gather with these devices that allows us to reinterpret scenarios and realities. And so now let's talk about the distributed component of this. I borrowed this from Bloomberg City Lab, very cool project on its own right, and sort of modified it for this conversation. So imagine throughout our environments, built environments and otherwise, we have these olfaction devices. And I actually intentionally don't call this an electronic nose because, keep in mind, we can have really sophisticated electronic noses, or we can have simpler ones that are really purpose-built for, really distinct measurements. And so let's keep it general as olfaction devices. And these can be throughout the built environment. They can be worn on the wrist of an individual, let's say in the form of a smart watch. They might be, on a scooter. They might be built into a car. And also importantly, some of these are stationary. Some of these are moving with a velocity. There's lots of rich sort of data that can be collected here. So how do we think about that in terms of an architecture that, really extracts the most value there and really delivers the meaningful augmented awareness that we're after? And so I like to think about, first the blue dot, again, that's an olfaction device, and that might be part of an edge device. Again, the edge device, it could be a smart watch, a smart boot, it could be, the smart toilet, it's whatever, but it's this device that's on the edge that is somehow interacting with the sample. And that edge device passes the olfactory data to some sort of networked device. But importantly, again, it passes the edge context data along with that. And then furthermore, the network device passes, of course, the olfactory data, the edge context data, but it also passes, additional local context data that may, again, be relevant to the interpretation of this olfactory data. And so let's say, let's go back to this example of, I'm an individual, I'm in my home, I have a smart toilet, and my smartphone is, let's say, making this API call to this database about, what's my prostate risk today, or what's my prostate trend rather. This API call, and it gets an API response that sort of informs that information. And that right there, what we have up right here is pretty powerful. It's obviously, more than what we have today in most cases, but I would argue it could be so much more powerful by including those additional data points, whether that's other olfactory information from other devices in your own home, or olfactory information from other devices throughout your community or throughout the other context. And so that sort of additional context is really super important to being able to make this meaningful information. So let's take all this sort of abstraction and make it like, you know, a little bit more concrete. What is my prostate cancer risk? That could be an API call. The response could be something specific. Stage 0.4 precancerous signals were detected. As a group, what percentage of a subpopulation X present with COVID-19 scent patterns over time period Y? 7.9%. Or as an institution, which children's parks are non-compliant with the city's code for generally acceptable odors and thresholds? Well, Irvington Park is unfit for child recreation. I want to point out a few things that's really necessary to take from this, from these examples. The first is that it's a misconception, and Andreas and others have really pointed to this throughout the conference. It's a misconception that answering the question with an olfactory data file is not what we're after. We're not really interested in the olfactory data file per se, except maybe in the case of, you know, OVR, where they actually want the olfactory data file to recreate the scent. But in most cases, we're really seeking the olfactory data file in the context of much more information to really answer the question. And the second point is, you know, to emphasize that it's super diverse, the questions we could ask of this information. You know, I was really inspired actually by Yuji's presentation earlier today about sort of the cross-species empathy and this psychological distance that we have. And how could, you know, how would the app that he would be developing, for example, retrieve this data and utilize this data to inform an augmented reality or virtual reality experience to achieve that goal? So those are really two key points. It doesn't have to be the pure data, and it can be used for extremely diverse uses. So we are a startup, and we do have an interest in this, and that is across sort of three different layers of this. The first is we are super interested in the fundamental electronic nose R&D, incredibly inspired by the work that Andreas and Shuguang and others have performed in this area. And this is sort of part of what, you know, how I got into this through my PhD work. We're interested in the recognition elements, and interestingly, we've also converged on GPCRs as a really critical factor to truly develop an artificial nose, and the functionalization chemistry and how that all sort of boils down into a fab process and manufacturing scale up and all the QA/QC questions that come with that. The second area we're interested in is actually the integration of that device and how that sample is acquired, how the gas or the volatiles are handled, and also what edge compute needs to be performed to get into that standardized file creation. I think earlier someone mentioned, you know, the .osm file, or I've thought of the, you know, the concept of the .olf file. Like, how are we going to make the JPEG of this data? And then finally, the connectivity between the edge device and the network device is an area of special interest to us. And, you know, thinking about passing that data over the wireless protocols and making sure that's compatible if you're doing NFC or RFID or Bluetooth or Zigbee or whatever it might be. And do you integrate that data on the network device, or do you actually intentionally want to keep that de-integrated until it reaches the lake house, so to speak? And of course, how that gets passed along. Those are some of our interests. You know, what is Aztec? What are we in terms of a startup? So we're actually sort of this bridge between sort of the pure academic world and the venture building world. And that is, we take what we find to be compelling R&D and IP. We might license that in. We might create some of that in-house. We work with what we feel are, you know, a really carefully selected team and really dedicated individuals. And then we sort of put those two things together and conduct extremely deep primary market research through a hypothesis-driven process that, you know, looks at macro market data, but we do a ton of, you know, person-to-person interaction and, you know, sophisticated analysis on the market to ultimately arrive at a thesis on what a fundable venture is, what the value propositions will be, what the key resources will be, so on and so forth. As Andreas mentioned, I am somewhat of a child of MIT, and we've taken that into this company. We're hypothesis driven. We really believe in the mens et manus philosophy of if you want to understand it, if you want to really make an impact, use your mind, but also use your hand. Really get your hands dirty. And we also benefit greatly from some of the amazing networks that are in the greater Boston ecosystem and elsewhere. And then I want to finally end on a note that, this has been a phenomenal conference to participate in. So much thought-provoking content here. And we would love to be able to contribute to any of these projects in any way possible, collaborate wherever possible, and continue to expand our team, whether that's the team inside of Aztec or just the broader network that we're operating within. So more than happy to connect with anyone who's interested in sharing some of these ideas with us. So with that, I'll pass it back. I hope I didn't go too far over.
Oh, that was great. Thank you so much, Joseph. I am also remiss because I actually screwed up the order. Sorry, Lucas was supposed to go, but it's all good. We're all getting friendly here, so apologies for that. And thank you. Please stay on. We're going to have so many questions for you. This was great. Now, for our next speaker, we have Lucas Lopez, and he is from Volatile AI, a Lithuania-based company, but is also, I think, global. We had the pleasure I had the pleasure of meeting him and his collaborator a while back, and we couldn't quite figure out where I fit and where they fit in our ecosystem, but now we know they're part of this family here. They're nosemakers, true, and they will tell us all about their technology over 5 minutes, and then I'm sure the people will have questions. Lucas, take it away.
Thank you very much, Andres, for the introduction, and thank you for inviting us to the conference. It's really great so far. So hopefully you can see my screen now.
Yes, we can see you and hear you perfectly.
Okay, great. So I call my talk Bringing Artificial Faction from the lab to the industry. And I wanted to share the story of Volatile as a company in which we departed from a technology. I started working at a research lab, and we are now looking for applications in factories and real use cases. So it begins back in 2010 when I was working at the Neurochem project in the University Pompeu Fabra in Barcelona, the SPECS research group. And basically I worked on this robot, which was navigating this wind tunnel. And the robot was able to find the source of a smell in the presence of distractors. So it had to identify what was the closest smell that it would find at one specific point. The main innovation at the time is that the robot was able to make classifications very quickly. So it would take a few seconds to make a classification, while an electronic nose would take minutes. Main innovation here was we were extracting temporal features of the signal by using a liquid state machine and representing them in a spatial pattern. So, well, these are two smell maps created from the robot. This is the top of the wind tunnel, and well, you can see that the classifier is slightly biased here, but that would allow the robot to find the right source of the smell 95% of the time. So that was a pretty successful project, and well, a few years later, I met Adomas, my co-founder, and we participated in this program called Entrepreneur First in London, and also in Hacks in Shenzhen in China. And after a few months of work, we came up with this device. You see here on the right. This is the device you mentioned before. It's sort of a handheld device in which you would point at something you would smell and press a button and would literally sniff and make a fingerprint of the smell. The device is on the left is the sampling device. So you have the nose on the tip of it, and it would help basically randomizing the sample. Not so interesting, but took a while to build, so I wanted to show it. So, well, that was the original version of the device. However, we realized that it was not really a good approach for the use case we're trying to solve. So we found primarily two big problems. First is that telling the difference between ammonia and ethanol blends, like my robot was doing, was fairly easy compared to tell the difference between two beers, which is quite subtle. On the other hand, the second difference is that variance in the data that we were collecting was phenomenal. And the reason for that is that this device was very simple, and that's something we liked, actually. But with simplicity comes a lot of variability in data. So many factors like airflow would affect the data, and temperature, humidity, useless things, but that would prevent us to really apply it. So after many iterations and work, months of work, we came up with a current version of the device. Many changes we wanted to avoid, but at the end of the day, we are pretty happy with this device. We trained it already to sample, to differentiate between different varieties of peanuts, coffee, and interestingly enough, asphalt. And we are getting pretty good results with this. We are more or less at the levels of human testing panels, so between 60 and 80% of an S-score. So, well, we thought we had solved the technical challenges, but then now the challenge that we are facing, I think, is the most important one, is more of a conceptual challenge. So the inner representation of the fingerprint that our device makes looks more like something like this. And I'm pretty sure all of you will be familiar with these radial graphs, in which you will have a set of abstract features telling the difference between the smells. Now, Adam has told me, like, wow, it's great to have this. However, there's a problem with this, and is that it's very difficult to sell it. And it's a fair point. The reason for it is that it's very hard to understand. So people would basically question, like, what does this mean? Is this related to chemical composition? Is this related to a panel tasting score? And well, we humans want to understand everything we see in front, and well, it's easier with other fields like images, but in the case of smell, it's quite difficult to represent it here. And we are working on that. We are running GCMS in parallel with our sampling. We are planning to run human testing panels in parallel, and we want to see how much we can infer one space from another. But in reality, we are aware of this body and brain tight relationship, right? And to illustrate that, I wanted to introduce you to this little guy. This is an Accidia. It's sort of a sea coral kind of thing, and it first days after it's born, it spends some time searching the sea floor, looking for a nip and quiet place to stick to. And once it sticks, it basically will remain there forever, not interacting much with the environment. So the interesting thing about this is that for navigation, it uses a very simple nervous system, almost like a rudimentary brain. But the thing is that once settled, it will literally digest its brain, right? And I think it's a perfect example of the concept I want to illustrate. There is no point in collecting and processing information of the environment if you are not going to use it. And most importantly, your perception is fully tailored for what you are and what you are planning to do with it. And well, how that translates into factories, right? Into applying a machinal function to factories. Well, the thing is that I remember Anne mentioning the important role of the observer in the system. I think the observer in this case would be the factory. So basically, factories do have raw materials. They take raw materials, they use industrial processes to transform them, and they come up with a final product. Now, these processes, they do have a set of parameters. These parameters can be tailored, can be tuned to optimize for energy consumption or waste reduction or final product quality. And the message, I guess, is that these parameters are the ones that should ultimately define what the smell fingerprint is for this system, and that basically a factory will have different perception needs than the ones of a human and or the chemical composition. And well, we believe that this is what has prevented machinal function to be widely used in factories until now, and we are working hard to fill this gap. Thank you. That's it.
That was just so great. And before I give it up to Carolina to introduce our next speaker, please do stay for the Q&A. I have to say that little organism you showed at the end, maybe you should put in the chat what it's called. How telling is it that once you settle and you're very happy in your environment, you end up digesting your own brain. You don't need new ideas anymore. It reminds me of a certain type of academic that finds their niche and never leaves. And I am happy to say that none of the people here follow that that modality. Nobody here has digested their own brain yet. So make sure you keep adding new experiences to your environment. Make sure you get out of your comfort zone, or you end up digesting your own brain. Okay, Carolina, please introduce our next speaker, who is I'm very happy to say we'll close the loop with our first speaker today, and because she's going to talk about user experiences. So I will let Carolina run the show until the Q&A, which will start after the speaker. Thank you.
Yes, so I would really warmly like to introduce Sara Nejad, who is a designer, and we started with Federico Castellano, and we talked about human-centered design, or as we established later, or like suggested later that we maybe should call it like life-centered design. So Sara is a designer connected to biocommunity, rooted in Media Lab in biotechnology initiative. And she's designer, she is collaborating and working at NYU, and she will be speaking about bioproduction of aromas to enhance flavors and food experience for astronauts. So this is like a very special user, but I believe very important. Please, Sara.
Hello, everyone. Hi, Carolina. Thank you so much for the introduction. And it's such an honor to be here among you all and have the opportunity to talk about my project. So I'm going to share the screen. All right, and hopefully you can all see it here. All right, so the project is called Aromata, and it's a concept for the biosynthesis of aroma compounds using freeze-dried cell-free systems in space. I'm sorry. All right.
Hi.
So a little bit about myself. I'm a New York-based artist and designer exploring the intersection of experience design, human-computer interaction, and biology to make sustainable innovations and innovate sustainably. And I want to give you just a little bit of background about how I got so obsessed with the future of food and food design for space exploration. During my thesis project at NYU about 2 years ago, I started to explore the relationships, the symbiotic relationships between humans, fungi, and algae. And I started to speculate different ways that we can grow these organisms for space exploration. And my research pointed to a few key findings. One of them was the importance of experience design in making better food experiences for the astronauts. And the other one was the potential of using synthetic biology to make these improvements. So what we know is that long duration space exploration is happening, is already in the works. And we know that space food is kind of not as, doesn't have the same quality that most astronauts would like to have. So there's this increasing need for new and safe, innovative ways to produce food in space. And we also know that eating and the dining experience is a very important communal aspect for the astronauts. It's the time that they all get together and share a meal. But as I mentioned before, the quality of food isn't that good. And to add to that, there is also this diminished sense of perceived flavors in space, which seems to be to the fact that the astronauts can't seem to be smelling the aroma compounds from food. And this leads to a decrease in their food intake, which is detrimental to their health. So how exactly is our perception of scent and therefore our experience of flavor related to our food experience as a whole? So flavor, what we call flavor, is this very complex combination of our perception of different sensory information, such as taste, smell, also the temperature of the food and the texture of the food. But among all these different sensory information, smell seems to be the more important factor. Another important and interesting fact is that we have a very limited number of tastes. So it's limited to sweet, salty, bitter, sour, or umami. But there are so many different aroma compounds. And the hypothesis is that we can combine different aroma compounds with a limited set of tastes that we have to create a more complex array of flavors. And by augmenting, so we can augment the flavor perception using aromas to enhance the astronauts' overall food experience. So to do so, we needed to figure out two things. One is, how do we actually produce these aroma compounds in space on demand and easily? And the second part is that how do we deliver these compounds, and what is the user experience for the astronauts? So currently, aroma compounds or flavorings are produced in three different ways. One is naturally, so through extraction from natural material, natural compounds. The second is the chemical synthesis of these compounds that are similar to the natural alternatives. And this seems to be the main method that aroma compounds are produced in the industry right now. And the third alternative is to biosynthesize aroma compounds using metabolic engineering and enzyme systems. So an added layer to this could be the applying the freeze-dry cell-free systems. So with cell-free systems, we make the complexities of metabolic engineering simpler, make it more time efficient, and make it an easier process to prototype and test. Also, by implementing a freeze-dried system, we can kind of condense the cell-free system with the DNA plasmids that are the instructions to how these enzymes are produced, and condense everything into freeze-dried pellets that the astronauts can easily combine with water and produce aroma compounds on demand. So introducing Aromata. Aromata is a design concept about a wearable olfactory device that allows astronauts to produce different aroma compounds easily on demand. It consists of two main parts. As I mentioned, the freeze-dried cell-free reaction mix is the main component that is then introduced to the wearable device. It consists of the cell-free extracts, the DNA plasmids, and substrates that are all condensed into freeze-dried pellets. These pellets are then combined with water, and the mixture is then added to the device. This process was informed by previous research done by BioBits, in which they use the freeze-dry cell-free system to produce different fragrant compounds. The second part of the device is the actual wearable component. It consists of a microfluidic chip, which is a bioreactor. It serves as a bioreactor. It also includes a control board, a heating device to make the bioreaction and incubation possible. And it also includes a piezo transducer to help kind of atomize these compounds from liquid to gas. And all of this is powered by a rechargeable battery. So all these components are miniaturized and placed in the back of the device. And then the nozzle is placed closer to the astronaut's nose. This design was informed by previous works about microfluidic bioreactors, and also the HCI work done by the Fluid Interfaces Lab at MIT, in which they kind of I'm sorry. In which they navigated different options for wearable olfactory devices. So the next steps for the project is to obviously design and prototype a complex aroma recipe. So going beyond one aroma compound and create the complexities that we seek to deliver to, for the astronauts. The second would be to prototype the device, then user test the device with analog astronauts, and then user testing in space. Thank you so much.
Wow, Sarah, an unbelievably scientific talk. I did not expect to see so much great science and engineering going on. I myself have quite a bit of experience with microfluidics. I invite you to check out Dr. David Kong's microreactors and cell-free expression that we tried to do with Shuguang also. Oh my God, is this actually a perfect segue into what I hope I understand to be Joe Davis's talk, which is coming up, and it will be the closing of our conference. Joe Davis perhaps will talk. We'll never know until he actually speaks what he'll say. But I feel that perhaps he will talk about the on-demand biosynthesis of compounds, of odorant compounds, by hijacking the ROSE system. He has already hijacked the ROSE system, which is connected to the Apple system, to create an apple genetically modified to contain almost all of Wikipedia. So he calls it the tree of knowledge. So if it is genetically encoded, almost word for word, the entries in Wikipedia in the genome of the apple. So he could, interestingly, to see if something happens to you if you eat that apple. So now it's the time for the Q&A, which has been, I want to say historically, even though it's been only 3 days for this conference. But what we've learned in this conference is that to have speakers speak for 5 minutes and then give the Q&A a longer time, we get much more bang for our buck. A lot more juices are flowing. So I would like to, Sarah says that she knows David already. So I would like to first present a question to each one of the speakers that has been coming in the roughly the order that they appeared, and then we'll open it up to the people. But if you want to say something, just raise your hand. And the first question goes to Shuguang. So Shuguang and his QTY code. I want to share an experience. When I first came to MIT, I was a postdoc. Before I became a research scientist with my own group, I was in Shuguang's group. And as a postdoc, I felt, on top of the world. I still do. I didn't know there was more to climb. But at that time, Shuguang wanted me to teach part of his class, but also to take his class. And his class was on structure of biomaterials. And he wanted all his students to paint with a marker or a paintbrush every one of the 20 amino acids. And I rebelled and I said, I don't want to do this. Why do I know mathematics? I have computers. Why should I paint anything? And I didn't. And I, to this day, regret it, because I think that Shuguang will tell us, but I think in engaging this other modality of the artist that I think every single human is born with, and we sort of beat it out of ourselves over the time. And some of you are lucky enough to have maintained it. That's how he thinks. Shuguang thinks in artistic terms in many ways. And how he came up with the QT code. Shuguang, how did you come up, and did art and design play a role in it?
Well, the QTY code comes from constantly ask questions. Yeah. So the question was, nature has already made hemoglobin, very water-soluble protein in our red blood cells, 30%. Nature also made the olfactory receptors, essentially the same alpha helices, is totally insoluble. Why and how? So as I said, God is in details, the devil is in details. You go in deep to see how nature made sense and learn from nature. And turn out this amino acids has striking similarities, and you can replace them and then become water soluble. But this has a lot of dead end. And the QTY is 3 amino acids. As I showed you earlier, the structure of remarkably similar. Just like I showed you the hand with the glove or without the gloves. It's the same kind of structure. And why come the code? Code should be simple. People have been doing this kind of work since the 1980s try to make membrane protein soluble. But use so complicated computer algorithm, use supercomputer to calculation. You put the same protein sequence into computer, each time they come out is different because the calculations not always the same. So you cannot, this kind of technology cannot spread to other places. The simplicity is the key. The genetic code, as I said, it's universal, it's simple. DNA code, ATCG, it's simple. Once you know one end, you know other one automatically. So we have come up with a simple code to do this. That's why we did. But we spent 7 years to crack the code. Initially, we were very hesitate to change many amino acids at once. It's called incremental and don't kill the protein. When you change 1% protein, that is 3 amino acids in 300 amino acid protein, most people said it's okay, you probably can do it. But if you change 5%, and in the room or 100 people, probably 50% people would say probably not working. If it's still then you change 20%, I would say 95% would say you're nuts, you're crazy. That's precisely what we did. So we are nuts, we are crazy sometimes thinking unconventionally, but follow nature's leads. That's how we made the code. And take 7 years. In China, we say take 10 years to sharp a saw, and here we take 7 years to crack the code. Genetic code, by the way, also take about 7 years. So that's
and there's also 7 transmembrane domains, the most conserved number in all of biology. I 2 comments on this. Since you mentioned the number 7, I want the audience to know it is an interesting mystery. Why is 7 transmembrane domains, 7 alpha helices, the most conserved number in all of biology? All of biology. I stand behind it 100%. And what I mean by this is, in every instance of any animal, any plant, anything, where we have sensing happening, the most conserved number is 7 transmembrane domains, no matter what you're sensing. You could be sensing light in your retina, which is the same exact system as the nose with only 2 differences. In the retina, the odorant is called retinol, and it's permanently stuck in the binding pocket of the rhodopsin, and then that, which is another GPCR, and that becomes an antenna tuned to different wavelengths by how the amino acids are constricting it. You tune the wavelength. Otherwise, it's the same system. The other difference is that it's biased backwards. The photons make it stop firing, while in the nose, the odorants make it start firing. So the 7 is the most conserved number, whether you're sensing light, smell, heat, pain, touch, toxins, you name it, mechanosensory, all of those. The Nobel Prize for this year for physiology and medicine was given for sensory proteins. So the field is heating up. The field of perceptual engineering is coming. And what do you think comes after you understand vision? Television. What do you think comes after you? Actually, not necessarily in that way. Television was first, then vision was understood. It was a case of reverse biomimicry. The other thing I want to say is that Shuguang's QT code is not random. He didn't just go ahead and change 15% randomly or shotgun it. He very precisely thought about it as a designer, I think, and as an artist. So for my next question, I want to ask something of Dr. Kolb from Aromics. So Ilya, you showed a fantastic slide of differentiability. And I want to talk to you about it for hours, but we don't have hours. So I have 2 questions. One is probably a yes or no, and is, have you tried mixtures of odorants on top of this? And the second one, which is kind of dovetails with this, is what happens when you invert this idea, where instead of trying to find the receptors that mostly correspond to this, and then you find ligands that mostly do the same thing. What if you try to, in fact, add one by one different things that will make the effect of the original ligand different? So those are the 2 questions: mixtures and sort of inversion of the system and the information flow in it.
Yeah, no, those are great questions, and it's something that's, it's 100% on the roadmap of what we want to do. It kind of goes with the scale up portion that I was talking about, where right now we just don't have the bandwidth to kind of do these experiments. I think so in terms of the mixture question, I think that's really interesting, because I think what we're finding is that and I'm sure, so I don't come from the field, so this is sort of brand new to me, but what we're finding is that, olfaction is very, very nonlinear, right? Which means that basically it could be that adding tiny amounts of one particular compound can cause either huge perceptual differences or no perceptual differences. And, I think that that is kind of at the core of what aromics is trying to quantify, right? So by basically building out the database, exactly as you're saying, of, adding things together and then seeing what the receptor level kind of activity is, and then correlating that to the perception activity, we can start to map out that space. And so in terms of inversion, again, that's 100% something that we want to do and just have not quite had the bandwidth yet. But it's super exciting that we even have the opportunity to sort of have this kind of assay system that works. And then once we kind of start to do these mathematical operations of adding and subtracting, I think that'll open up the field quite substantially.
Yeah, thank you for that. So my next question is for Professor Lee, Wen-Yee. It's going to be a little bit different than the others because I have a personal connection with Wen-Yee and we're working together on a startup. And I want to ask her, as an academic person who is delving into business, what have you found to be the challenges and the opportunities? What works, what doesn't? Because I see you as an example of lab to market transition. And this is what academia is for us to keep writing papers to talk to each other is not going to do much for humanity. What Professor Lee here is doing is taking a sabbatical from her day job to create something for the planet. So I want to know what she has been experiencing, what she likes, what she doesn't like, and what is her advice for others who want to follow in her footsteps?
That's a very good question. And I remember when I start to think about how to get out of my comfort zone, because it's very intimidating, to say the least. Okay, one advice that I got from the instructor is get out of your lab. So get out of the building. So to answer your question, Andres, I think that a lot of time we've been the researcher in academia, we sort of like using our brain a lot and putting in the lab, and we only talk to the students or people who are in the same field. But whatever great idea we may have, it comes with a question. This is great, this is wonderful, but is this something that people really need now? So I'll give you an example. Earlier, I showed that our approach can do kidney cancer and also can do prostate cancer. So we're saying that at the end of the day, we're going to develop a model that can detect all different cancers, blah, blah, blah. And then when we talk to one of the manager at a hospital, they say, but we really don't need that. And I was like, what? I'm giving you the best of the whole world and you're telling me you don't need it? And then they say, no, because if you are telling me that this patient has 10 cancers, we still have to run 10 tests. So what is, and so at the end of the day, what we have found, or what I come to realize, that we really do need to talk to real people to know what they need, and then from that we can really fine tune our research to a point that really benefit people directly. Because what we're talking about right now is really something for application, if you will. Right, commercialization is an application. So if you come up with the best product in the whole world, but you're the only person who want it, then yeah, you only have one customer. Yeah, so that's what I learned. Get out of the building. Yeah, I still remember this this remark. Just get out of your building, talk to real people, and yeah.
That's such great advice, and I think it'll stick into everybody's head. My next question is for Dr. Azzarelli. Joseph, there's two questions for you actually. One is very simple, and it says, but also deep. Can we fossilize smell? And the second one is a bit more convoluted, which I think I would like you to talk a little bit more about your ideas around the monetization of scent. and the scent data stream, or as Carolina today baptized it, she christened it today Osmo data. I think we should all marry that term. It's a fantastic term. She came up with it on the fly. I love it. Osmo data, because sense data is not a good term. Osmo data. So the monetization of Osmo data, because you hit it right on the spot. You said many times, you said decentralized, distributed. The fact that we can get this, something that the animal world cannot do, by the way, each nose is localized. The power that will come from distributed sensing is so vast. It is like trying to see the birds, but then you don't make jet engines out of feathers. This is what's going to really transform it. So the two questions is, can you fossilize scent or Osmo data? And tell us more about the monetization of the distributed scent data.
Great questions. Let's start with, the fossilization. So this is a super interesting question. And I think that this is something that's really fundamentally tricky or distinct about this sort of dimension of information. And that is that, from my really fundamental background is synthetic organic chemistry. And one of the things that you learn is that all organic compounds transform over time when they are not at 0 Kelvin, basically. And if they're exposed to light or heat or oxygen or other reactive species, they will all slowly transform, or transform at least at different rates. And so I'm actually going to answer this question with an example. We took a deep dive into breath analysis for lung cancer screening. And one of the most interesting and frustrating things that we learned was the sensitivity and specificity was correlated to how quickly we could get the breath sample into either the GCMS or the electronic nose. And that's because some of the compounds we were looking for were fundamentally short-lived once they left the cell that was producing those compounds. And so I think that that's sort of really illustrative of the fact that, I would be skeptical that you could fossilize a smell. And this is actually why I think it's super brilliant what the Aromata, Sara's project, where you're actually just the genesis of the smell in real time when it's needed. I think that makes a whole lot of sense. So anyway, going back to then the second question, which sort of relates to the first question actually, which is, you capture the scent profile at a moment in time, and that's super critical, that timestamp, because again, it's going to change over time. The monetization. So this is something I think that actually parlays back into the past 2 days, which is, this is data that is being uniquely generated by us. We have rights to it. We should have rights to it. And if we want to obstruct its use, we should have that right. And if we want to monetize its use, we should have that right. And so I think that there needs to be some thinking around this Osmo data or these .osm files. I'm not an expert, but it seems to me that this should somehow maybe leverage the blockchain or leverage other ways where the integrity of the data can be preserved, and the individual who has rights to the royalty stream cannot be disintermediated from that data. And so I think that there needs to be a lot of thought there around, how can individuals be monetized or monetize their data if they want to? And I think this has really profound consequences on 2 fronts. The first is, guarding against unintentional use of the data or unintentional outcomes of the technology, which is obviously a major theme here. And the second is, I actually think that building in this feature into the data set from day one is actually a major incentivization to build the database. When people know that, hey, my excrement smell has value, or my bad odor has value, or whatever that is, it's sort of like the beginning of crypto, where there was this actually incentive to be contributing and computing. Same thing. I see a potential to have very strong incentivization for early participants into this data concept. So I hope that was helpful in answering those questions.
That was indeed very helpful. I don't know if you can hear me. I've lost sound for some reason. Oops. Somebody say
We can
something.
hear you.
Okay, then. I can't hear anyone else. But my next question is for Lucas Lopez and Volatile AI. And again, it's a two-parter. First of all, I want to congratulate him on being, I think, the only one who spoke at length about tracking. He had a tracking robot. And all of the questions I've been pouring in is about, oh, can you track another? Why can a dog do it? We had Stephanie Brenner from Noam Sobel's lab talk to us about the challenges of putting an incontinent which adult needs diaper change, and it's because of directionality. My internet connection is unstable, so maybe I don't know, will take over in a minute. However, I want to ask Lucas to talk a little bit more about tracking. And the second part of the question, and I'm sorry to throw two at you, but I might lose connection, is why did you go after peanuts? Peanuts is such an interesting thing because of the allergies, right? Can you detect peanuts in food at trace concentrations? And in that way, you can be saving lives tomorrow. So please talk about tracking and peanuts.
Right. So, well, we moved to peanuts because, well, you know, our first use case was beer. Beer turned out to well, we all love it, but it turned out to be very complicated to smell, right? It had problems with the humidity. CO2 was changing constantly. It was oxidizing lively. It was a very changing product. And it was basically very challenging. Also, ethanol was creating a very big signal in the sensors. So seeing very subtle differences to the point in which customers would need was really difficult. Then yeah, we moved to peanuts and yeah, coffee and asphalt. Just, well, we had the opportunity. It's more of a dry, more less changing product. So it was really welcoming for this application. We haven't really tested if we can detect peanuts inside a mixture with other things. We were focused on seeing the difference between different varieties for now. But that's really a good point. And
I can just see your device being installed in every kindergarten in America, and people have to pass through it to make sure that they're not bringing peanuts. You know, people are elsewhere in the world making fun of Americans and Western Europeans, but it's a true terror. People are dying off of things that we weren't yet dying 15 years ago. Babies are born so allergic to things that one bite might kill them, and it's a serious threat. So I see an immediate application of your technology to just scan every damn food that somebody's bringing in, just like we scan for guns at the airport.
True. No, that's actually true. We had this incident recently, in not at the office, but basically with some friends, and we had to run to get the medicine precisely because of this at our restaurant. So yeah, very true. So and first question, sorry, was about tracking, right? So.
Yeah, yes, tracking. You showed a robot that could track and find the source of an odor. I've often been asked about this, and I would love to have next year we'll have Boston Dynamics here, and maybe we should have a sniff off. Will their robot find the source of the odor faster than your robot?
Sure. So that robot was well, that was long ago, right? But the key point of it was to make very quick classifications. So many of the times you would get false positives, but it was following the basically what the behavior moths do. So basically, moths, when they are looking for a mate, are making this zigzag movement until they find a trace of a pheromone that they are interested in. At the moment they have the trace, they would fly against the wind. And this is precisely what the rover was doing. It would just do zigzag until it would get a good signal of something that I would call it its attention. And then it would have this wind sensor, and then it would just run against the wind. And yeah, that was very interesting. We were trying to apply this into a drone, a flying robot. Of course, the turbulence of the rotors would just mess it up. But yeah, that was basically the inspiration of it.
Yeah, that's great. So we have one final question from me for Sara, and then I'll let Carolina take over and see if there's more questions from the audience, or if we're going to go to talk to the students. I know their input. So for Sara, again, a two-parter. First of all, do you know of anything from the astronauts about how bad is it? How bad is the smell on the ISS, and how do they do it? Do they habituate? And what is the shock of someone who just boarded? Is it hit them in the face? And secondly, you spoke of a very interesting device that you would have for on-demand. It's basically conscious choice of which aroma you want to experience. I wonder how often most people do that. Most of the time, I think the choice of aroma is unconscious. You walk into somewhere, boom. Few of us use perfume consciously, and I'm at fault of this. I should do it more consciously. I don't do it enough. So please talk to us about how bad is it on the space station, and what are your thoughts on the conscious choice?
Sure. So I haven't had the chance to speak to any astronauts directly. What I know is from just secondary research. And yes, you're correct. There is a problem with odor. There's a problem with body odor that I know, like one of the propositions at the church lab is to kind of make the astronauts less prone to produce body odor. There's also another problem with the fact that any aroma compound that is produced kind of lingers in the air, and because it's a confined space, it doesn't really get out. So this, the fact that we have this mixture, complex mixture of different odors lingering in the air, that also makes it more complicated and difficult for the astronauts to, if they want to really enjoy the experience of, let's say, having coffee or vanilla ice cream and really like just focus on that, that capability is diminished for them. So for the design of this device, this is to your second point. For the design of this device, well, at first, like I was even considering like, what is the best method to deliver these compounds, since this is a communal experience, and should we design some kind of universal food aromas that can be matched with a lot of the different kind of foods that astronauts are having, because they don't have the same exact menus, they have personalized menus and food. So is there a way that we can have this universal smell, and then they can all enjoy it, and it could be a shared experience? But again, through my research and really looking at the problem that you don't really want to produce that much smell that will take up the whole space, that is a problem. So that's why I looked into personalized devices and wearables that produce just enough amount of smell that is detectable by the person who's wearing it. And that's why when I looked at the paper published by the Fluid Interfaces, and they looked at different ways that we can actually accomplish this, and their results pointed to a necklace, something similar to a necklace that hangs, that seemed to give the best results. It was not as detectable by other people, and it also produced enough scent to be perceived by the wearer. However, because of the altered gravity in space, I kind of like intuitively, I thought, okay, wearing a loose device around your neck would not be the best method. So with this device, I thought I kind of looked at what are the devices that astronauts already wear around their head, and the device resembles something that they already wear and are used to wearing. And also, it has the capability to be fit around the neck as well. So you can wear it around the head, behind the ears, or around the neck. But it fits well. It's not loose, and that kind of addresses the problem at 0G. So I'm not sure if I answered both questions.
Yeah, I think that it's interesting, and it's very interesting that you also mentioned that George Church wants to deodorize the astronauts. Yeah. Well, that's one way. Another way is to make them anosmic. Yet a third way is to find little fungi or other things that can live inside the spacecraft and consume these odorants as part of a filtration system. In fact, we have a whole project with Chris Maurer, who I linked his video on his site earlier, which was inspired by NASA, and it ends up now in Namibia. So space research, such as what Sara is doing, doesn't necessarily have to go to space before it brings benefits to humanity down here on Earth. So your work is an example of that. I think it will benefit humanity down here much faster than it will benefit the astronauts. But thinking about space and thinking about these new environments is very generative of new ideas and new technologies. And everybody talks about Tang being invented for astronauts, but that's the least of our things here. The stuff that Chris Maurer is doing that came out of the NASA NEAT project. Someone told us, well, if you're proposing mushrooms from Mars as a building material and as a way to compact human waste, again, about the human excrement. This olfaction conference has concentrated quite a bit on human excrement. Yeah, if you thought, somebody said, if you're talking about putting this on Mars, why don't you show first that it works in Namibia, where the climate is arid, but not as bad as Mars? And indeed, we went, and 3 years later, we're building houses in Namibia right now. We're growing mushrooms. We're thinking about deploying it as filters to filter out water from mines. And I know what perhaps you don't know is that there's a thing called radiotrophic fungi. Most people don't know about this. I didn't know about it 3 years ago. And these are fungus that actually thrives on consuming ionizing radiation, gamma rays and X-rays. They can be used to protect astronauts while at the same time providing food. And on top of everything else, they grow like flowers grow in the direction of the sun. They grow in the direction of the leak, of the radiation leak. So if you have a spacecraft that's traveling and it's been bombarded, the mushrooms will actually show you where the leak is and add mass in front of it, therefore automatically preventing the leak from getting worse. It's quite amazing. This is my last question. There's many more that have come in, but I'm going to pass it on to Carolina in just a second, after I remind everyone that in 24 minutes, we have the closing speech here by what many have called the most interesting artist alive, Joe Davis. And he is a bioartist and he is a scientist also, and he works at both MIT and Harvard, and actually a couple of other universities too. And he's world famous, and we're so honored to have him. He will start speaking at 3 o'clock. Between now and then, we will enjoy the productions of various students and others who have contributed to this. And I want to thank the speakers for this session so much. Thank you for your contribution to this conference. I would very much love to see you again next year, and also remain part of this community. I know that Carolina and the rest of the team here are very keen on having a LinkedIn channel and a Slack channel, and people throwing their ideas at us through the chat has been so generative. And I am in awe of this conference. And Anne-Sophie Barwich, Professor Barwich, says that my excrement now has value is the quote of the day, and I disagree. The quote of the day is, this is the conference I've been waiting for. That's the quote of the day and the quote of yesterday. So with that, I'm giving it over to Carolina. Please take it away.